Okay, welcome everybody to the About the Data webinar. Just want a couple of logistics. This webinar is being recorded. We will be posting the video on the GDC website under support shortly after it's processed. If you have any questions, we just ask that you hold them to the end and place them in the Q&A section of the web conference. I am Sharon Gaheen. I'm the Technical Project Manager for the Genomic Data Commons Project from Frederick National Laboratories. I'll be giving an overview of the data that we have in the GDC. My colleague, Bijou Isaac, is our scientific lead from our Frederick National Lab supporting Leidos biomedical research. So let's go get started. This is the agenda for today's webinar. I'm going to talk about the sources of data we have at the GDC. We provide access to about 65 projects, and they roughly cover 68 different primary sites. So I'll be giving that overview. Bijou will be talking about the different types of data that we have at the GDC. And then I'll be ending this webinar with a feature presentation focusing on how to access the data in the data portal and just some useful areas in the GDC website, such as our publication pages and our data source pages. Before we get started, I just want a couple of notes. I'm going to focus on the data that has already been released in the GDC. So that means if you go to the data portal, you'll see the data there. We do release data roughly every 2 months. We have a data release. We have an upcoming data release, data release 27, which is due out in late October, early November. And please join our Twitter or check our release notes for all the latest version of the data and new data that's submitted every 2 months. The GDC, we make data available for research purposes. We have a bunch of programs that we support at the NCI, which are listed here, as well as programs that donate data to us. And we greatly appreciate the data that's donated to us from many different organizations. A lot of the information you'll see today is on our data sources page, which is on our website, which I'll be reviewing later. We're going to kick off with TCGA, the Cancer Genome Atlas, which probably needs no introduction. It is certainly a pivotal program. It transformed our understanding of cancer, and it led the groundwork for many programs and projects, such as the GDC that we have today. It was launched in 2006 by the NCI and NHGRI. It characterized over 20,000 primary samples, both tumors and normals, with 33 different cancer types. Roughly, TCGA generated over 2.5 petabytes of genomic, epigenomic, transcriptomic, and proteomics data. Numerous publications, as you see in this image, resulted from TCGA. The 2018 publications focused on the PanCan Atlas Consortium, which looked at understanding how, where, and why tumors arise in humans. The GDC maintains these publications, and we'll be going through them later on today. I did want to also congratulate the TCGA team, if any members are on this call, for winning the 2020 AACR Team Science Award for a job well done. Going into the TCGA data that we have in the GDC, we have roughly 11,315 cases, and you can kind of see here the case counts by the different data categories and each of the different projects. Each of the different projects are by disease type. So as you can see, BRCA, we have a lot of data from TCGA BRCA, which is breast cancer, GBM, which is glioblastoma multiforme. We have a lot of different data from the different projects, and that data includes the clinical biospecimen, slide images, the alignments, somatic mutations, expression quantification, copy number, and the methylation data. One thing I want to note too is that users can go into the GDC and explore the TCGA genes and mutations, because we do make the somatic mutations open access through our exploration tools, and we'll be showing them a little bit later. The top graph is just an example of the top mutated cancer genes within the TCGA program, but you can certainly use the GDC to drill down and see those genes and mutations by the actual projects that you select. Going on to Target. Target is a collaborative effort of a large consortium. It is managed within the NCI by the Office of Cancer Genomics, which is OCG, and the Cancer Therapy Evaluation Program, which is CTEP. It does use resources from the NCI's Children's Oncology Program, which is COG, and also does strategic partnering with SPECS, which is strategic partnering to evaluate cancer signatures. Target is in phase 4 of the program, so we're in the publication area. The project teams within project are separated by the different disease types that you can kind of see here. So we have ALL, AML, NBL, osteosarcoma, and renal tumors. The project team PIs and the COG lead, they lead the clinical trials associated with that. And the goal of these project teams are to accelerate molecular discoveries for the hard-to-treat cancers and also rapid translation into the clinic. As you may know, that despite the survival increase of 20%, despite the survival increase in cancers, still 20% of pediatric cases do not respond to therapy. And so we have to look at the current treatments, and also those the current treatments do have a lot of harsh side effects, and they do affect development and growth. And so Target has been geared towards understanding the treatments. One thing to note too is that while you see here in this particular publication, we still see about adult mutations that are absent in younger AML patients. One of the trans target initiatives, the studies looked at 142 driver genes and only 45% matched adult drivers. And you can certainly read more about Target on the OCG program and access the publications on the GDC publication page. About the Target data we have in the GDC, there's a little over 6,000 cases. One thing I want to point out is I have these numbers here, the PHS numbers. If you're not familiar, to get access to control data in the GDC, you go through dbGaP, and these numbers are the dbGaP accession numbers, and Bijou will be going into later how you can apply for access to some of the control data in the GDC. A couple of things to note about the Target data. In the GDC, we have the project IDs that are pretty much mapped to the disease types that I showed in the previous slide, but there are 3 phases to Target ALL. And so what you see is P1 is phase 1, and that's the pilot phase that focused on 200 high-risk B-cell ALL cases. Phase 2 focused on the early bone marrow relapse, and phase 3 focused on acute leukemias of ambiguous lineage, which is ALAL. Going into the Cancer Genome Characterization Initiative, or CGCI, this project focused on rare cancers, and there's 2 primary programs, projects that the GDC supports from CGCI. There's the Burkitt Lymphoma Genome Sequencing Project, which is BLGSP. This was a joint initiative between the OCG and the Foundation for Burkitt's Lymphoma. And if you're familiar with Burkitt's, it's a type of non-Hodgkin's lymphoma with a chromosome translation of the Mike gene. And the goal of this project was to explore the genetic changes that lead to prevention, detection, and treatment of this rare and aggressive cancer. There's still a lot of treatment success has been variable, and so their focus is on understanding those genetic changes. And the second project is the HIV Tumor Molecular Characterization Project, or HTMCP. That's a joint initiative between the OCG and the Office of HIV and AIDS Malignancy, which is OHAM. And they're looking at the genetic events that drive HIV-associated cancer. You know, why certain cancers have a higher incidence in HIV-positive patients. The cancer that we're currently supporting in the GDC is a cervical cancer. It's the 3rd most common women cancers worldwide. And the goal of that is to look at potential therapeutic targets. They look at HPV infection, obviously is necessary, but it's not sufficient for tumor development. And what this group did is they looked at clades of cervical cancer cohort and compared them with TCGA. They did a functional enrichment analysis of the differential expression genes and found 170 HPV subtypes that were organized into clades, with alpha-9 and alpha-7 being the most carcinogenic. So that's the CGCI program. In the GDC, we have, like I said, the cases from cervical cancer, about 212 cases, and from the Burkitt's lymphoma, we have about 120 cases. You can kind of see the different types of data that we have in this graph below. And for the most part, we have the alignments and the expression and the splice junction quantification. Going into HCMI, HCMI is a collaborative international consortium that focuses on generating tumor-derived culture models. These include 3D organoids and 2D conditionally reprogrammed cell. And if you're not familiar with organoids, organoids are sort of their miniaturized versions of organs produced in vitro in 3D, and they're often used for drug screening and to study the tumor microenvironment and for response prediction. And this HCMI was established by the US NCI and teaming with the Cancer Research UK, which is CRUC, the Wellcome Sanger Institute, and the Foundation for Hubrecht Organoid Technology. HCMI developed models and related data that's available as a community resource. There are 4 cancer model development centers within HCMI. These include Broad Institute of MIT and Harvard, Cold Spring Harbor Laboratory, Stanford University, and Weill Cornell Medical College. These groups are generating the models. The models are searchable through what I'm showing here is the HCMI searchable catalog, and they're also distributed by the American Type Culture Collection, the ATCC. There's about 17 of these models, 17 cancer types associated with these models. For the HCMI data in the GDC, we have 23 cases, but we are getting additional cases that are currently under review. And the available data includes the clinical data, somatic mutations, gene expression data. One thing to note that was special about HCMI is when we were developing support for it in the GDC, we did have to extend our data model to support the concept of a model as a type of sample with a child-parent relationship to both a tumor and a normal. And so that was one of the special things we did to support HCMI. Another thing is users can go into our exploration tool, the genes and mutations. Somatic mutations are available as open access, and you can view those through our exploration tool, and I'll be showing those a little bit later. DLBCL, this is actually a study that looked at identifying genetic subtypes of DLBCL. There's actually 2 particular projects associated with that. About over 500 biopsies were studied. The 2 projects are the NCI-CCR genomic variation in diffuse large B-cell lymphomas, or NCI-CCR DLBCL, and there's also an NCI clinical trial sequencing project that focused on the DLBCL study. So these 2 projects came together, and they worked on algorithms to discover genetic subtypes based on the co-occurrence of genetic alterations. There were 4 prominent subtypes that were identified: MCD, BN2, N1, ECB, and these are available on the GDC publication page. There was a publication created in the New England Journal of Medicine that had those. So the GDC, we have about 534 cases from when you add up the 2 different projects which form the study. And we have different data, such as sequencing data, alignments, gene expression, and the splice junction quantification. CPTAC, the Clinical Proteomics Tumor Analysis Consortium, is another program that we support. CPTAC is a nationwide effort that was established by the Office of Clinical Proteomics Research, and they apply proteogenomics, which is generating the proteomics data and the genomics data, and looking at characterizing the tumors based on that data. They also aim to generate both data and assays to make available as a public resource. There are 2 particular programs within CPTAC. One is the CPTAC Tumor Characterization Program, and they're focusing on characterizing the proteins and genes to better understand the molecular basis of cancer. And what you see here is this, in the top area is the CPTAC Tumor Characterization Program teams. They consist of 3 what we call PCCs, which are the Proteome Characterization Centers, and 5 PGDACs, which are the Proteogenomics Data Analysis Centers. And the PGDACs are focused on doing analysis of the data that's generated by the PCCs. The other project is the CPTAC Translational Research Program. and they aim to understand drug response and resistance to therapies in the context of a clinical trial. And the three cancer types that these translational research teams are focusing on are ovarian, AML, and breast cancer. And you can see the different organizations involved in each of those as well. In terms of the data we have at the GDC for CPTAC, we have about 990 cases of patients with diverse disease types. When you go to the GDC, they're organized into two projects, CPTAC2 and CPTAC3. CPTAC2 was an initial project that focused on breast, colon, and ovarian cancer, and CPTAC3, the disease types were expanded to include brain, head and neck, kidney, lung, uterine. These are the ones that we support at the GDC. We have lots of available data for CPTAC and the GDC that you can see here. But a couple things I want to talk about is for CPTAC, there's a public data dissemination, in addition to the GDC, which supports the genomic data as well as the clinical data. CPTAC also has a proteomics data coordinating center that submits data into the Proteomic Data Commons. So both clinical data and proteomic data are made available in the Proteomic Data Commons. Imaging data from CPTAC is made available in the Cancer Imaging Archive, or TCIA. And then there's a CPTAC assay portal for distributing the targeted assays and an antibody portal for distributing the antibodies. So both the data and the assays are being distributed. The Multiple Myeloma Research Foundation, or MMRF, is a nonprofit organization that was set up in 1998 to find a cure for multiple myeloma by looking at innovations and that accelerate the development of treatments to extend the lives of patients with multiple myeloma. The one project that we support in the GDC for MMRF is the Relating Clinical Outcomes in Multiple Myeloma to Personal Assessment of Genetic Profile Studies, which is known as COMPASS, and that's MMRF's landmark personalized medicine initiative. One thing to note that's special about this is it is a longitudinal observational study. There's about 1,000 diagnosed myeloma patients that receive standard and approved treatment, and the vision is to track this treatment every 6 months. These patients are followed for over 8 years, and so the longitudinal data is something that is important and is special to the MMRF that we have in the GDC. The GDC provides, we have about 995 cases from the COMPASS study. 111 of those cases have sequencing data from 2 time points. And there's the clinical data, the alignments, the somatic mutations, and the expression data are available. And we do make MMRF data, the genes and mutations, also available through our GDC Exploration Tools. The next program is the Count Me In, and this is a nonprofit initiative for patient-partnered cancer research. It's run out of Broad Institute of MIT and Harvard and the Dana-Farber Cancer Institute. It was launched as a patient-partner cancer research project where anyone in the US and Canada can share samples and also their voice to accelerate discoveries. There's over 8,000 patients that have registered, and there's 6 ongoing Count Me In projects in the GDC, six ongoing Count Me In projects with 2 in the GDC currently and more planning to launch soon. So the GDC right now, we support the Metastatic Breast Cancer Project and the Angiosarcoma Project. We have the data in the GDC. Patients go to the CMI site, they register online for the CMI study. They fill out a brief survey on their cancer experience, and then they can consent to share their saliva, their blood, and their FFPE tumor samples, as well as their medical records. And then the Count Me In program shares the integrated data sets, which include the patient-reported data, abstracted clinical information, whole exome, and the RNA-seq data. And it's shared in a pre-publication manner in order to expedite research. So researchers can harness the CMI data from different repositories like the GDC to better understand their cancer research. And more information, if you're interested on the Count Me In, you can go to joincountmein.org. And if you have any questions specific to Count Me In, then please feel free to go ahead and contact them directly with any questions. So the Count Me In data that we have in the GDC, we have about 200 cases from the metastatic breast cancer study and 36 cases from the angiosarcoma study. And we do have the alignments, somatic mutations, gene expression, and splice junction quantifications in the GDC. Beat AML is one of the largest data sets of its kind for leukemia. This is led by Oregon Health and Sciences University to better understand drugs that should be prioritized. There's 11 academic medical centers that are accruing 900 AML patients, and there's about 11 pharmaceutical and biotechnology companies that are supplying drugs for testing. The 900 AML samples are undergoing functional screening, and then after the functional screens, there's computational modeling and target validation to look at demonstrating the drug effects on the target to provide so that an overall therapeutic benefit can be provided. Clinical trials are conducted on the validated targets, and what they're looking at is mutants that are sensitive to drugs and drugs that are affecting mutants are identified. There's whole exome and RNA sequencing data that's captured. There's also ex vivo drug sensitivity analysis that is performed. If we go and look at the data in the GDC, we have 583 cases for the Beat AML cohort study. And the cohort study is the functional genomics landscape in AML. And there's 56 cases from the crenolanib study. And this looks at the resistance to crenolanib in AML due to diverse molecular mechanisms. And so we have clinical data, biospecimen data, the alignments, and also the whole exome somatic mutations available from Beat AML. One thing I wanted to note about Beat AML is they also have drug sensitivity data that's available in the Cancer Target Discovery and Development Portal, the CTD2 portal. And there's also a nice mining tool, visualization tool to mine data sets known as Visome. Another effort is GENIE. This is an American Association of Cancer Research, or AACR project. It's an international PanCan registry. There's data sharing of about 19 international cancers with a goal of improving clinical decision-making. This is an ongoing clinical sequencing efforts that's CLIA and ISO certified, and it's serving as an evidence base for the entire community. Within the GDC, we have over 44,000 cases of data from GENIE version 5.0. And it's organized into 8 sites. So when you go into the GDC, you see like GENIE MSK, which is Memorial Sloan Kettering, DFCI, which is Dana-Farber, and you can view the different data by the different sites. A couple things with the GENIE data, the mass somatic mutations, copy number scores, and transcript fusions are provided. Clinical data from the GDC Onco Tree, what we had to do was to map the data from the Onco Tree to the GDC Data Dictionary. We work closely with our clinical curators, work closely with the GENIE team to do that. We don't have the raw alignments for the GENIE data, but the genomic data was lifted over from HD19 to the GDC reference genome standard, which is HD38. Foundation Medicine is another program that we provide support for, and it's a molecular information company that specializes in precision medicine. And they have a standard genomic profiles that is used. It's called Foundation One is their genomic profiling platform. And they routinely analyze cancer specimens using the advanced sequencing that they have in Foundation One. For the FMI data, the GDC has over 18,000 adult patients across 23 different disease types that you see here. We have clinical data by specimen, and we have short variants with substitution and indels. One thing about FMI is we do make the mutations available through exploration, but as controlled access. So a user does have to have controlled access, have authorization to access the data. Those were some of the primary components, programs that we support. There are more as well. The OHSU-CNL program, there's about 176 cases of chronic neutrophilic leukemia. And This includes, whole exome and RNA-seq data. We also have data from organoid-pancreatic. We have about 70 cases associated with pancreatic cancer organoid profiling. And this is a valuable resource for pancreatic cancer researchers because they can use it to compare primary tissue to organoid culture. And they did find that the organoids really do recapitulate the mutational spectrum of pancreatic cancer. The RNA sequencing did show the presence of transcriptional subtypes. The VeraPop Apollo, we do have a small number of cases associated with bronchus and lung cancer from the Research for Precision Oncology program that established this cohort for the Veterans Administration. I just want to say something about Apollo, which is the Applied Proteogenomics Organizational Learning and Outcomes Network. This was a collaborative network between the NCI, the Department of Defense, and the Department of Veterans to start incorporating proteomics data into the patient care. So we do have a few cases from them. And the WCTD MCRCP effort, which is the West Coast Prostate Cancer Dream Team Genomic Characterization of Metastatic Castration-Resistant Prostate Cancer. The goal of this particular project is to illuminate the molecular mechanisms of resistance to therapeutic agents in the treatment of metastatic castration-resistant prostate cancer. And lastly, I did want to highlight the Cancer Cell Line Encyclopedia. We make, at the GDC, we make the cancer cell line data available on our legacy archive. And these are culture cells that can be propagated in order to look at the genetic and pharmacological characterization of these models. I want to kind of highlight these because these are alignments that are open access. And so if there's organizations that, we do get contacted for, whether there's open access alignment files such as BAMs to be used in some of the different tools. And so these are open access. So what I provided was an overview of the different data that we have in the GDC from the supported cancer programs and data that's been donated. But there are many programs that the GDC supports, such as Exceptional Responders, Alchemist, and Match, in which the data has not been released yet. And our upcoming data release, which is DR27, we are going to have some new gene fusion data and also whole genome variants from our Sanger pipeline. So that's some new data types that are coming soon. That's an overview of the data sources. Now I'm going to pass this off to Bijou to give an overview of the data types.
Thank you, Sharon. In this section of webinar today, I'll be talking to you about the GDC data types in general. Next slide. First, to talk about the GDC available data. GDC supports and provides access to many different data types, including clinical, biospecimen, and genomic data. Once the data is submitted to GDC, it is standardized and users are provided access to it through the GDC portal. Data types such as core clinical and biospecimen data elements are standardized across projects, while data types such as sequencing reads are aligned to a common reference genome, for example, the HG38 build. Higher level genomic data such as gene expression quantification and somatic mutations are generated from GDC standard pipelines. Details about these pipelines can be accessed from prior GDC webinars on GDC bioinformatics pipelines. The figure on the right, commonly referred to as GDC data model, shows how the GDC data is organized. I'll not be talking about the details of this crowded figure, which is also available on GDC website, but as you can see from the legend, GDC provides data files, analysis files, index files, metadata, notations, biospecimen, clinical, and administrative data. Individual data elements within this model are standardized and defined in GDC Data Dictionary, which can be accessed through the link provided on the slide. Next slide. To talk about the GDC data types, the category of the data types available in GDC and their associated file formats. Clinical data such as demographics and diagnosis are provided as TSVs and JSONs, while supplement files are provided as TXT and XMLs. Biospecimen data such as sample and aliquot information is provided as TSV or JSON, while diagnostic slide images are provided as SVS files and supplement files as TXT or XML. For genomic data such as the whole exome sequencing, aligned reads are provided as BAM files, while simple somatic mutations are provided as VCFs or MAFs. BAM aligned reads are also available for targeted sequencing, whole genome, RNA-seq, microRNA-seq, and ATAC-seq. As with whole exome data, SSMs are provided as VCS and MAFs for targeted sequencing and whole genome data types. Targeted sequencing data additionally contains copy number variation data as TXT files and transcription data as TSV files. RNA-seq data type additionally has gene expression quantification files as TXT and TSV, and splice site junction quantification data as TSVs. In addition, GDC has recently started providing gene fusion data in BED-PE format. MicroRNA-seq data types are the microRNA expression data quantification and isoform expression quantification data in TXV and TSV formats. Among the other data types, GDC also makes available methylation scores from methylation arrays and copy number scores from SNP6 genotyping arrays. For ATAC-seq, GDC only provides aligned reads as BAM files. Next slide. This slide shows the organization of clinical and biospecimen data within the GDC. Within clinical data across projects, each individual case has information on patient demographics, diagnosis, follow-up, exposure, and family history. The diagnosis data has another level of information on follow-up, treatment, and pathology details, while molecular test information is provided within the follow-up. Within biospecimen data model on the right, subset of GDC data model is shown as shown before. Each sample is shown in a parent-child relation with aliquot, analyte, portion, and slide data type. Also, aliquot data is related to read group data, while portion is related to analyte. In addition, some project-specific data elements are also provided by GDC as clinical supplement files. All clinical and biospecimen data is available for download in TSV or JSON formats. Next slide. Slide images. GDC also provides access to diagnostic and tissue slide images. The image on the right is a screen snapshot of a particular slide image. These images can be interactively viewed on GDC portal with zoom function. File properties table above each slide provides information on each individual data slide image. These images can be downloaded from the portal in SVS format. Currently, there are slide images from 32 TCGA projects, with more in the planning from other projects in future releases. Next slide. So before we talked about files available for various data types. In this slide, we talk about files specifically available for various genomic experimental data types. These are among the most accessed data types from GDC other than the clinical data. As you can see from the extensive list, these data are available for users in various file types. The first table provides the list of file types for whole exome and targeted sequencing, which are among the earliest data types in GDC. These experimental strategies have minimum number of have been generated from the maximum number of GDC pipelines, and therefore they have a variety of file types available. These include the BAM files from BW Aligner, somatic mutation data from 5 variant calling pipelines. They are available as VCF or the variant call format files. Additionally, mutation annotation format or MAP files are also available that contains aggregated mutation information from VCF files. In addition, the mutation stability index sensor metadata is available from tumor-only data. Most genomic files are sensitive data, and therefore all such file types are that potentially contain germline information are protected and distributed through a controlled access part of GDC portal. For the whole genome sequencing, files available from GDC include the BW-aligned BAM files, somatic mutation calls from Sanger pipeline in VCF format from 2 different workflows, Pindle and Caveman. Again, the MSI sensor metadata from tumor-only data, copy number segment, and gene level copy number files from ASCAT pipeline. Structural rearrangement data from BRAS pipeline are available in BED-PE and VCF formats. For RNA-seq experimental strategy, star-aligned BAM files are available. Transcriptome-aligned genome-aligned, and aligned chimeric BAMs are available. So there are 3 different kinds of BAM files available for RNA-seq. GDC has the gene expression quantification data in TXT format from HT-seq and STAR pipelines. These are primarily counts, FPKM, and FPKM upper quartile from HT-seq and the STAR workflows. Additionally, GDC has gene fusion data in TSV format from the 2 pipelines, Ariba and STAR Fusion. For microRNA-seq, we have the expression quantification data in TXT format from the British Columbia BCGSC pipeline. MicroRNA-seq and ATAC-seq file types also include the BWA aligned BAM files. Next slide. Next, I'll talk about the 2 of the important file formats in GDC, which is the VCF and the MAPs. For the uninitiated, these can be complex files. Within the GDC, VCFs are produced from 5 different variant calling pipelines when comparing a set of mass tumor normal alignment. The figure at the bottom provides general information on variant calling workflows. Each of the variant caller produces its own VCF files, which can be of 2 types: a raw simple somatic mutation VCF, which are the unannotated, and the annotated somatic mutation, which are the annotated with pipeline-specific information on somatic variants. The mutation annotation format, or the MAF, is a tab-limited text file with aggregated information from VCF files and are generated at the aliquot level through the somatic aggregation workflow. MAFs are produced by aggregating the GDC annotated VCF files generated from 1 pipeline for 1 project. MAF files in GDC are of 2 permission levels, open and controlled. Next slide. To talk about the variant call format, GDC does follow standards on file format. So GDC VCF is a version 4.1 specification. At the bottom of the slide is an example of an obfuscated VCF file for viewers. A VCF file format contains a meta-header section on top and a variant information section. The meta-header section is at the beginning of the file and has information on workflow, sample and reference, and starts with a double hash symbol. This symbol allows the different VCF file reader softwares to recognize and differentiate between information content. The variant information comes after meta-header section, and each row represents 1 variant with chromosomal coordinates, a reference and alternate alleles, etc. The info field within each row annotates the variant. Next slide. Mutation annotation format. The GDC map file format follows the TCGA map specifications with additional columns. The figure at the bottom of this slide is an example of an open access map file. More information on GDC map formats can be provided on the link provided for the GDC doc site. It's given below. The map file provides information such as total analyzed samples, submitted IDs, date the file was generated, and the GDC file version, and also the specification version information. Each row in map table below the header section provides information on a particular variant call, such as Hugo, gene symbol, entrance ID, sequencing center, reference build, chromosomal coordinates, and stand information for these genes. Translation effect of the variant allele, such as the missense or whether it's silent, variant type, whether it's a SNP, insertion deletion, or dry or tri polymorphism, etc., reference and tumor alleles, or whether this variant is reported by other databases such as DBSNP with RSIDs available. Otherwise, these are classified as novel. As with VCS, GDC map files are of 2 access levels, open and controlled, and depending on the information content in the map file. Next slide. Next, I'm going to talk about how to access these GDC data files. As mentioned before, GDC makes available for its users both open and controlled access data. Open access files are clinical, biospecimen, whole exome mass somatic mutation, gene expression quantification, etc. While the controlled access file include aligned BAM files, raw and annotated DNA-seq mutations, such as VCF and MAPs. In addition, individual supported programs may require project or program-specific data use restrictions, and that those need to be adhered to. The figure on the right shows how the access controls on GDC works. In general, GDC requires and validates user authentication through ERA comments and DP-GAP authorization to access controlled data. Once the users have access to a research project through DB-GAP, they can allow users within their group to access same projects through their DB-GAP account. For those interested, the link at the bottom of the slide can provide more detailed information on how to obtain access to controlled data. With that, I'm going to hand it back to Sharon for a section on GDC feature demonstration. Thanks, Julie.
Thanks, Biju. Okay, I'm going to go into demonstration of the GDC and how to access data. This, if you're familiar with, is the GDC data portal. And the first thing we're going to do is we're going to go into projects. And this is where you get information of what data is in which project. We're going to go ahead and select a program. We'll select TCGA. And what you can see in here when we select TCGA, you see all the projects associated with the TCGA. You see the different cases that you can then select and the different types of data that we have and available files. You can also get a nice distribution of cases per project up here and look at the top mutated cancer genes in the selected projects. If you go to the graph view, this is where you see that, what I had in the data source presentation. You get a nice graph and an understanding of which projects within the TCGA program have different types of data and the number of cases that are available for each of the different data categories. If you go back to table, I'm going to just go ahead and select the first project, TCGA. This is considered the project entity page in the GDC. It gives a nice summary information about the project. It gives you a link to the DB-GAP study accession number if you want to apply for access to the different projects. It gives you a nice overview of the cases and file counts by the data category and also by the different experimental strategy. One thing to note is up top, this allows you to download all the biospecimen data and the clinical data associated with the project. You can also download a manifest file, which, if you're familiar with the data transfer tool, which is what we have at the GDC for downloading large groups of files, you can then pass in that manifest file and use the client tool, the data transfer tool. We do have an extended video on the data transfer tool on our GDC website that you can certainly watch if you're interested. So this is how you get project information. I'm going to now go into exploration. Exploration in the GDC is where you go to further explore a project and also build a cohort by looking across different projects. So with exploration, what you can do is we have what we call faceted searches for cases, clinical genes, and mutations. I'm just going to go ahead and select kidney here. And when I select kidney, I can see there's 2 programs and several projects, TCGA and Target with kidney, with data associated with the primary site, kidney. I can then go in and look at genes associated with kidney. I can look at the distribution of most frequently mutated genes. I can also look at an overall survival plot. For that, I can choose a particular gene that has a mutation and also select here the survival, so I could see the survival on whether, like in this case, I've selected VHL, whether VHL, the survival for cases that do not have a mutation versus those that do. I can look at, drill down and look at detailed mutations as well, and you can further drill down into specific pages associated with that. What I wanted to primarily do is just kind of show you how you explore across projects here. This is our OncoGrid, which gives you a nice heat map type view of the genes and the mutations and the cases across what you've selected, focusing on the 200 most mutated cases and the top mutated genes by SSM. But you can also filter by specific mutations and also by copy number changes as well. So this is another way to get access to data, but in exploration, you build a cohort by looking across the project. You can also save this cohort. So if you want to do analysis later, you select to save the cohort. You could save this as a gene set. I'm just saving as a kidney cancer gene set. And also you can also save the cases as a new case set. And if you have attended our other webinars on analysis, you'll know how you can use those case sets in the analysis. I want to go next to the repository page. When you go to the repository page, this is where you can view and download the data by selecting many different faceted search, including projects. In this example, I'm just going to show, we go to Files, and I'm going to show that you can add we'll add masked somatic mutations. We have about 3,500 of those. And this is pretty much how you add data to the cart. You can select here, you can pull down this cart and select add all files, or you can add individual files. I'll just add this to the cart. And then when you go to the cart, you can actually download the data associated with the cart by selecting cart, or you can download the manifest file and use the data transfer tool for downloading it, which is very important for large data sets such as whole genome BAMs. You want to be able to download them efficiently. You can also download biospecimen data from the cart, clinical data from the cart, and a sample sheet that links some of the samples to the different the clinical data that we have. So that's downloading a masked somatic mutation. The other file type I wanted to show you, because we are focusing on projects in this webinar, the other file type is, I'm going to go to select clinical, and I want to note there's what we call supplement files. Supplement files are additional metadata that the project wants to give the users, and not just, we support many different types of clinical data and biospecimen data at the GDC. Sometimes there's project-specific data elements that they'd like to share, and those are in clinical supplement files, and they can be, shared in XML, TSV. There's this biotab format, which is a nice text file of some of the clinical data. But if you wanted specific, clinical data that are specific to a project, you might want to take a look at these clinical files and download. You can certainly select them here. You can download it here or add them to a cart and download. The next thing I want to show is in repository. This is a nice feature that users aren't always aware of. What we've done at the GDC is we've looked across projects and programs, and we've created a pretty large data model that allows you to search across, do cross-project comparison. But we certainly don't have every single data element, on the faceted search, because it would be very, very, very long. But we do have this ability to add the case and biospecimen filter. So if you go to our data dictionary, and if there's a field you want to that's in our data dictionary that you don't see in a faceted search, you can simply type it here. So for example, I'm going to choose a sample type, and when I select it, it's added here on the left in my faceted search. We do get a lot of questions on people that want tumor and versus normal, and this is how you do it. You once the custom filter is added, then you can go ahead and search for your type, your particular sample type. I'm going to go ahead and select to add another one called sample composition. Oops. See. Sample composition, and this, this sample composition, you can kind of see if you want to get to data on organoids. This is a nice way to search, because I've talked about organoids from HCMI and also from the Pancreatic Organoid Project. Well, this is a way that you can look at sample composition. Similar to the case and biospecimen filters, that you can create these custom facets, and you can just exit the custom facets by selecting the X. There's also a file filter here that you can do. And so for file filter, I'm going to go ahead and select workflow, because we do get questions on, when somebody downloads the data, what's the workflow type that's used. So you can see here I've added, a file facet on workflow, and so you can see a particular, if you want to search for a certain workflow within the GDC, like Star Fusion data, you can select that and you can get access to the data. So I just want to point this out, because this is a really useful feature to get to project-specific information that you may not see, apparently in the facet. So that's a brief overview, a demo of the GDC portal. I wanted to go to the web page right now, and This is our GDC webpage, and if you go to support and webinars, you'll certainly see information on the webinars, obviously, and the videos. But if you go to About the Data, what you'll see is an area called Publications. If you're not familiar with this, this is the GDC. We support a bunch of publications, and we make the data available for download from those. So I'm just going to go ahead to Publications, and I'm going to select PanCan Atlas, which is what I referenced earlier in the presentation on TCGA. And you can see there's 22 publications from PanCan Atlas that we have at the GDC. What you can do is you could then click on a publication page we have at the GDC. If you click on the title, that'll go directly to the publication. But here are the data that we have in the GDC for this publication. You can download the manifest file of the open access data. If you can download it for the controlled access data. If you have, if you're authorized to dbGaP to do this, you can use the DTT then to go ahead and download the data. There is instructions down here for downloading using the DTT. You can then access the different files individually by selecting them. And these are supplement data that is not searchable in the portal per se, but it was data that was made available through the publication. And so that data is available in the GDC. Another area of the GDC is the data source pages. The data source pages, hopefully the information I provided today, it gave an overview of the different programs. It is available if you go down here and look at the different data sources that we support. And so, for example, if we drill into CPTAC, what you can see here, you can get an overview of the program. But there's also useful links. We have, if you want to click on here, this will launch the data portal, the GDC data portal. There's also proteomics data within CPTAC and the proteomic data commons as well. And here's where you can get links directly to the different applications that the different programs use. We have, information on the cancer types that you can click directly on that to launch, that particular cancer type. And then we have listed here the data types and the different access levels that we have for the data types with links that query the GDC data portal to get access to that data. As you can kind of see here, it just is a nice query. So you might not be familiar with its data source pages, but I just wanted to let you know that they are there. Okay, and so that was the demonstration. I just wanted to put up this references page to show you what I showed today was the publication pages, the data source pages. A video of this webinar will be made available once it's processed. The data dictionary is available on our documentation site. And if you want to look at the different fields, you can then use those fields by in our custom faceted search, which I demonstrated today. The data release notes are available here. Every 2 months, we typically release data. Bijou gave an overview of VCF and MAF file, and you can get more information on the formats there. And lastly, support. Please feel free if you have questions that are associated with the GDC or data to contact our support or and join our user listserv as well for more information on data releases. And follow us on Twitter because we do advertise our data releases on Twitter as well. And that's pretty much an overview of today's webinar. Does anybody have any questions? Okay. The one question we have is, will the slides from the presentation be available afterwards? We are making the video available on our submission, or I'm sorry, on our GDC website. So if you go to our GDC website, we just here, go back to our website. This is a nice little breadcrumb that allows you to get to the different resources we have at the GDC. If you go to support, and if you go to, support monthly support webinar, you'll kind of see what we do is whenever we finish processing the videos, we make them available through this page. And so they're taking a little bit of time to come up, but you'll see the videos there from each of the different webinars. We're still processing some of the videos because we do have to take time to edit the captions, but the slides will be made available as a recorded video, after we finish processing. Are there any other questions? Okay. All right, it looks like we don't have any other questions, but thank you very much for attending today's webinar. Much appreciated, and you'll have a great rest of the day. Thank you, everybody.